All right. Okay. Well, everybody who is here has record permission, I think, hopefully. You could give that a try and yeah, see. All right. All right, well, yeah, weather again. You know, of course, I think it's beautiful outside, but on the other hand, there was an official winter weather advisory from the National Weather Service because of road conditions. And then, even though it's kind of nice, I think, right now, the wind is supposed to pick up. And even if we managed to get the lab done in 2 hours, by the time we were scheduled to hopefully finish lab, the wind chill would be down in the single digits. And I think that these things kind of combine to me to indicate that it's not particularly safe to do lab for everybody. I know some of you would be totally fine, but I think not everybody would have been. And plus, when it's that cold and people are uncomfortable, it's hard for people to learn, when they're really cold. So with the midterm exam coming up next week, it seems like it's simply, sadly, regrettably, a better use of our time to have class in the classroom. I think I told you in the beginning of this semester that lab would occasionally have to get changed because of the weather. I posted a heads up last night on D2L and emailed it, and then as I told you I would, I made the decision before 9 a.m. this morning so that everybody would have a chance to change their plans as necessary. So you're all here, so you clearly got the message. Hopefully the people who are not here also got that message. And maybe they will show up. All right, well, what we have for today is I have some review topics in mind that we're going to cover to kind of wrap things up constructively, pull things together for this first unit of the course. So what I have is, let me see here, this is going to work. Let me just change one more thing out here. Out in D2L, I'm going to create a Limnology. There we go. All right, so we have several topics that I would like to cover with, just checking here. Okay, one person has emailed me that they'll be missing class. Okay, so we have a couple of different topics to cover in this review session, and I think that what I want to do is start right off with the first of them, which will be evolution and Hardy-Weinberg. Since we went over that on Tuesday of this week, it's kind of fresh in your minds, but perhaps not the Hardy-Weinberg aspect of it. Now, I'm sure that you have all taken genetics, pretty sure that you've all taken genetics, whether that is a unit within general biology or an entire one semester genetics course kind of at the sophomore level, which is now a required part of the biology major, although it is just an option for students in the environmental science major. So let's consider the Hardy-Weinberg example that was covered in the reading. I'm going to walk through this, and then I'm going to give you a problem or two to solve. All right, so let's going to consider, this is an evolution topic. But let's consider a population of 1,000 individuals for whom that have a particular locus with 2 alleles. And there are 360 individuals in this population that are genotype AA. There are 480 of genotype AB and 160 of genotype BB. And that's, again, this is the example in the reading questions. Actually, I think it's also the example that's worked through in the textbook. So forgive me if you're actually totally prepared and already did this, but I want to just review the vocabulary and the procedure here. Now, the questions that we want to know about this particular population are, what are the alleles? What are the genotypes? And if there's phenotypes associated with those alleles, those genotypes, what are the phenotypes? And we want to know, what are the frequencies of these things? So the first thing I'm going to do is I'm just going to check to make sure that this adds up to 1,000. 8 and 12 is 20, 7, 8, 9, 10. Okay, adds up to 1,000. 1,000 individuals, there's only 3 genotypes. It must add up to 1,000. All right. So let me ask you these questions. You know, what are the alleles? And their frequencies. And similarly, what are the genotypes and their frequencies? Now you could do this in words if you're a word kind of person. I'm a kind of a graphic kind of person. I like a table or something kind of graphic thing. And I'm going to show you a table, a type of table that's just really useful. And the way the table works is like this. You put the alleles over here, and you put the genotypes here. And if you have phenotypes, you would put them over here. Now, in this example, we have 2 alleles, A and B. And we have 3 possible genotypes. It's a diploid organism. So it could be AA, could be AB, or it could be BB. And we don't have any phenotypes in this example, but if there were phenotypes, I'd put them over here on the right. If it was some kind of a complete dominance kind of situation, there'd be 2 phenotypes. If it was incomplete dominance or codominance or something else, then there might be 3 different phenotypes. Okay, so we want to figure out what the frequencies are of these things. And what we're given is the genotypes. And there's 1,000 individuals. And of those 1,000, 360 of them are AA. So that means 360 out of 1,000 is the frequency of the AA genotype. In other words, 0.36. I think you could probably fill the rest of these in, and I'll fill those in while you do that too. AB's 480 over 1,000 equals 48. 160 out of 1,000 equals 1/6. I'm just going to make a little check up here. 0.36, 0.48, 0.6. Now there's only 3 genotypes. So if these are the frequencies of them, you know, which is like the percentages, right? 0.36 is 36 hundredths. 36 hundredths is the same thing as 36%, you know, percent per hundred. So this is 36%, this is 48%, this is 16%. And that's it. There's only 3 alleles. I mean, there's only 3 genotypes. Sorry. So this must all add up to 100%, or else I made a mistake, right? So let's see. 6 and 6 is 12, and 8 is 2. 7, 8, 9, 10 is 100%. Okay. So these are correct. Now from these frequencies of the genotypes, you can calculate the allele frequencies. And the brilliant conclusion, inference reached by Hardy and Weinberg separately, mathematicians, was the realization that if all of these things, if these genotypes are all reproducing equally well, there's no mutation going on, there's no migration coming in or going out, they're all surviving equally well, and they're not, you know, doing anything special, mating only with each other that are similar or whatever, any of those things, then the frequency of the alleles is going to be very simple. Oh, well, actually, it's even simpler than that. The frequency of alleles is really simple and continues to be simple. Okay, so what's the frequency of the alleles? Well, look, these 360 individuals each have 2 copies of the A allele. So there's going to be 720, because they're diploid, right? So there's going to be 720 copies of the A allele. Held by these individuals. These individuals, every one of them has 1 copy of the A allele. So these individuals hold 480 copies of the A allele. In other words, when you look at the alleles held by these ones and these ones, the AA individuals and the AB individuals, that group of individuals holds 7, 8, 9, 10, 11, 12, 1,200 A alleles. Now the total population is 1,000 individuals. If every individual is diploid, then there's a total of 2,000 alleles at this locus. 2,000 because every individual has 2 of them, and there's 1,000 individuals. So there's a total of 2,000 alleles at this locus. And 1200 divided by 2000, what is 1200 divided by 2000? What percent is that? Anybody there? Yep, 60. That's right, 60%. So this is 60%, or in other words, 0.6. Just kind of highlight that, put a box around it so you can see that. Now you can do the same thing for the B alleles, which is to say these individuals, there's 160 individuals, each of them has 2 copies of the B alleles, so these individuals are holding 320 of the B allele. There's 480 of these individuals. Each of them is holding 1 copy of the B allele, so that's another 480 B alleles. You add those 2 together, you get 800. 800 Bs out of a total of 2,000. And what's 800 divided by 2,000? Or 8 divided by 20? What is this converted like to a percentage? Chat there? Yeah, so what percent is this? 800 out of 2,000. What percent is that? Yep, 40%. Now there's only 2 alleles, right? There's only 2 alleles, A and B. And so the sum of the percentages must equal 100%. So just to check that, let's see, does 60% plus 40%, yep, that equals 100%. Okay, so that just means we didn't make a mathematics error, you know, some kind of little arithmetic mistake in here. It adds up to 100%. Now, this seems pretty obvious, really. If there's only 2 alleles, the frequency of them has to add up to 100%. I guess I think that's pretty obvious, you know, because if they didn't add up to 100%, there must be another allele out there that you weren't counting. So if there's only 2 alleles, it must add up. Those 2 must add up to 100%. Now, like I said, that seems really obvious, but it's kind of amazing that people actually think that's not obvious. I mean, because they memorize it in class as if they couldn't figure it out every time. What I mean is this frequency here is called P. P is the frequency of one of the alleles, little p, and Q is the frequency of the other allele. And so when we say that those 2 have to add up to 1, what we're really saying is that P plus Q has to equal 1, 100%. Well, I think that maybe you would agree with me that's not particularly profound. It doesn't really reveal anything about the population, except that there's only 2 alleles. If there were 3 alleles, P plus Q wouldn't equal 1. It'd be P plus Q plus R equals 1. And if there were 4 alleles, P plus Q wouldn't equal 1, but P plus Q plus R plus S, or whatever you want to call them, would have to add up to 1. So this is where this rule, P plus Q equals 1, P plus Q equals 1, comes from. It's just a statement that there's 2 alleles and their frequency adds up to 100%. Okay, so you know, this is the beginning, the basis of the Hardy-Weinberg what we, the sort of the Hardy-Weinberg problems in biology. And tell me if you've learned this before. If you've learned this before, give me a little check mark. If you're like, oh my God, this is the first time I've ever learned this, give me an X mark. Okay, so almost everybody responded, and everybody who did respond yes. Okay. Well, what I've got for you now is, I just wrote this right before class today. So hopefully it's all good. Yeah. What I have for you is, if you go to the D2L site, you will find a, what do you call it? A folder? No, a thing. And it's called For Today. Here, I'll show it to you. I can do that. So go out to the D2L site. And it should look like this. And here, I gotta get. I get Jeffrey in here so I can see it. So go out to the D2L site. There is in material content, there is a new folder called For Today, the 4th of January. And in that folder, there is a worksheet. I'm using that abbreviation WKS to refer to worksheet. So what I'd like you to do is download that worksheet and take a look at it. And I'm going to let my Jeffrey into this meeting. There's so much going on here. All right. So what I'm going to do is I'm going to have you join into a breakout room. I'm going to put you in a breakout rooms and have you work your way through this worksheet as a group. Now everything in this worksheet is basically review, and I'm asking you to review and apply it to an example there. Now we're going to have 4 breakout rooms. If you would like me to join your breakout room because you have a question or you want some help, you have a little button, I'm pretty sure, unless they removed it from this version of Zoom. Anyways, you should have a button that looks like this, a blue question mark, and it says something like, I don't know, help or something. All right, so download a copy of that worksheet. Appoint someone to be the chair of your group. Appoint someone to be the note taker. Now, the note taker, you could work on this copy of the file electronically and just save it and then save that as a worksheet. There's probably other ways you could do it too. I don't know, there's some other ways you could do it, but you want to try to work out your way through this worksheet, putting the answers in it, and then I'm going to have we'll share them as a class and go over it. All right, so every group needs someone who's a chair who understood these instructions. And some kind of note taker, I think, is a person who can pull up a copy and share a copy of the worksheet. All right, do you have any questions about this assignment? Okay, so use this as a...
I have a quick question, Virginia.
Sure.
Since I just got here, I've been waiting for like 20 minutes.
Really?
Yeah.
In the waiting room?
Yep.
Zoom only told me a minute ago that you were in the waiting room.
Really? Because I've been there since 1:08.
No way.
Yeah.
Okay. So
can I just ask you really quick what we're doing?
We're going to review today, and what I did was I walked through Hardy Weinberg, which you've had a class from me before where I think we did this. Yeah. And now I have a worksheet for you, which is on D2L, and I'm going to put everybody in breakout rooms to do the worksheet.
Okay, sounds good.
Yeah. Yeah, somebody else yesterday said they were in the waiting room for a long time.
I feel like Zoom's been having issues because on Tuesday my audio wasn't working and it kept crashing on me. It is what it is.
It is, yeah. We just keep, anyways, I'm glad you're here.
Thank you.
Yeah. All right, so papers here. Okay, so any other questions before? All right, so I'm gonna send you to the breakout rooms and have you work your way through this worksheet, and it'll take a little while. And this is your chance really to discuss it with other people and try to figure things out so that you really know how to do this. Okay, so here's some breakout rooms are now created. And let's see, get some people assigned to rooms. There's going to be a little weirdness. Okay, has everybody got a room? Okay.
Handy dandy calculators. So
in the table. Fill this table.
the population 10,000 version.
Can you guys hear the music in the background? Do you want me to turn it off?
Can't hear a thing.
Yeah, I don't hear it. You're good.
So that's 9,000 dominant yellow.
So that one is from the 90%.
Huh?
That one is the one came from 90%. That one, 9,000.
I can't hear what you're saying. The one for what?
Can you hear me now?
Yeah.
The one from 90%.
90%? Yeah, is the
Okay.
yellow. And then blue is the
Okay.
100. So there's no yellow.
Yeah, there's no There's no mixing yet.
Do you guys know how she did those calculations? It's like she,
For the other part?
yeah, because it seemed like she doubled one of them. So you would say,
You know, I think for the part that's in the middle, where you've got
yeah,
the 9,000 and the 1,000, you might actually want to put that over on the yellow and the blue, just to say the population. Because the 9,000, like the part where you've got the 9,000, the middle part would be the genotype, and so that would be your percentages. So then I think for there,
okay,
you just put in like 90% and 10%. Because that's what her table looked like.
yeah.
Or you know what though? You're right. She did double them.
Yeah, like for the alleles, so that's like what goes here.
Yeah, she doubled those because there's 2 copies of alleles per, what's the word I'm looking for? 2 copies of alleles per individual, right? So yeah, it'd be 18,000 and 2,000, I think.
And then she added them up, and then she divided by.
So I think that's gonna be 20,000 or 2,000.
Yeah, so it would be the 18,000 would be divided by 20,000 in order to get your genotype frequency, which is just that 90%.
Okay.
So
By
I don't think
2,000.
you have to worry about the math for this part, just because you already have the percentages.
Yeah, it's all just going to be 90%, 10%.
Yeah.
Y is 18,000 out of 20,000, which is 90%. And B is 2,000 out of 20,000, which is 10%.
Oh,
In the absence of the 5 things that cause evolutionary change, what will the population be like next year?
So wouldn't it be the same?
Yeah.
Just because nothing, yeah.
I think this is where the p squared plus 2pq plus q squared comes into play for this next one here.
Yeah, what we learned in biology. I'm trying to go ahead and put.
Because if it was the same, then we would have the exact same ratio, and we would have no hybrids, right? But like some homozygous are going to breed with some other homozygous of the other type, and then create hybrids, right?
Yeah.
So actually, I don't think it's going to be 90, 0, 10% for that.
This is now, so like I'm just putting what was in the last one, and yeah, like we got to figure out next year with this part here. Yeah. Alright. You guys want me to pull up that chart? Let me see if I can find it. This one? This one? Oh, so like we just do a Punnett square of like what we have. It's really not hard. So we have yeah, so it'll be heterozygous capital Y and lowercase b. Oh wait, but there's no, but there's 1,000 of them, 9,000 of them, so there's more. Oh,
So so this is where the p squared, that whole Hardy-Weinberg equation comes in. So p right now is 0.9. That's our y, right?
yeah.
And then q is going to be 0.1. So p squared, 0.9 squared is 0.81. So that's going to be our yy, right? Then So
Say
yeah,
that one more time.
so that's going to be 81% because that's p squared. Then
If the
2p.
formula is p square plus pq plus
No,
q square.
p squared
Okay.
plus 2pq plus q squared.
Oh, 2p q square
So
equal to one or 0?
yes,
Equal to 1 or
should
0?
equal 1. So
1.
in
Okay,
this
I got it.
case, p, yeah, so let's see. So p times q, 0.9 times 0.1 is 0.09, and then times 2 is 0.18. So it'd be 18%. So yeah,
Sounds good. Said
our
18.
our YB would be 18%. And then BB would be Q squared, which is 0.1 squared, and that's 0.01. So 1%. And then we just figure out the rest based on those numbers.
So is it still at 10,000? Oh, is that?
So if yellow is dominant, then we're adding that to, like we're adding the YB to the YY. Is that correct?
Yep, that's how I understand it.
Okay.
So in this case, 81% plus 18% is 99%, right? So 99% out of 1,000 is 9,900, right? Or I'm sorry, was it 99 or yeah, 9,900. And then minus 1 0 there. And then the other one would just be 100.
Hey, room 3, how's it going?
It's going.
Yeah, it's going. Okay, that's good. Oh yeah, I see some numbers. I see some numbers there. Looks like you're doing good. Do you have any questions for me?
Yeah, can you explain how you got this part again? You double
Well,
the population and then you add this what's the second number?
yeah. So you have calculated the genotypes, and then you look and you see how many Y alleles are there now in the population. You could assume, you could assume that there's 1,000 individuals if you like working with numbers rather than percentages. So if 81% of the population is YY, that's 810 individuals. Each of those has two alleles, two Y alleles. And then so you would write that over there in the Y box. And then you got your 18% that are heterozygous. 18% of 1,000 is 180. So you got 180 Y, yeah, from that one. Oh, yes. So every one of those 810 has two, yep. And then you add those up and divide by 2,000, and that gives you your percentage. Yep. And so 1,800 divided by 2,000. And you can do that in your head, or you can do it on a calculator, or, yep, 0.9.
So where do the 2000 came from?
Well, if there's 1,000 individuals, and each one of those is
Where
diploid,
then?
then the 2,000 comes from 2 times 1,000.
Okay, so I think we have 10,000 individuals.
Oh, in the
For a specific,
population? Oh, you're right. It's 10,000. Yeah. So it would just be that everything multiplied by 10. It's still going to give you 90%.
Okay.
Yeah. So instead of 1,620, it would be 16,200. Instead of 180, it'd be 1,800. But I think the key point is to notice that once again, it is 90%. Yeah.
And so, kind of in the absence of the things that cause the Hardy-Weinberg to not work, then the population would just remain static after that, right? Because P and Q no longer change.
Exactly.
Okay.
So here in the second generation, or year one, whatever you want to call it, because the P and Q are exactly the same as they were before, the frequencies of the genotypes are going to be exactly the same as they are here, like you said. So you guys just keep working through this worksheet. When you're done with the worksheet, just leave the breakout room and come back to the main room, and then I'll know you're done. And I think that when the first group is done, then I'll stop and get everybody to come back and we'll go over it. So it's not a race, but when you're done, come out to the main room. And if you need me, press the blue button. Any questions for me?
Thank you.
Okay. All right. I'm going to go on to the next room.
What was that formula again?
That was p squared plus 2pq plus q squared. So because p and q didn't change, everything's going to be the same as it was last year.
So yes, it is in Hardy-Weinberg equilibrium for year 0. So would you guys say yes?
Well, yeah, it is in the Hardy-Weinberg equilibrium. But for our numbers, are they supposed to be, like, is there supposed to be any mixing then, or no? Like, did she, did she tell us that it was supposed to stay the same for the 3 years that we're looking at it? Or is it changing?
So for the first one, obviously it's going to be different, but after that, yeah, it's just going to be completely the same because it's at equilibrium. The ratios will no longer change after that because p and q aren't changing. So the calculation, the equation, p squared, all that is
Right.
just going to give us the same result every single time. That's kind of the equilibrium.
Oh, okay, gotcha.
I think the big clue is about the 5 things that cause the evolution she mentioned above. Because when there is no, in case of Hardy-Weinberg equilibrium, there is no mutation and any other cause of evolution. So we can say it is in Hardy-Weinberg equilibrium.
Can you say that one more time?
So could you pull? I need to see the one she wrote on the upper one. Could you go up once? Okay, no, just down, just a little bit down. Right here, in the absence of 5 things that cause evolutionary change, what will happen to the population be like next year? That means the 5 things that cause evolutionary change, if you guys remember, it's a mutation, it's genetic drift. Those, in case of Hardy-Weinberg equilibrium, there should not be mutation, genetic drift. Those are, I think, the criteria for Hardy-Weinberg equilibrium.
Yeah,
And this,
so in the
yeah.
absence of those things, yeah, you're right. Nothing changes. So yeah, it just remains completely static, and that's what the equilibrium is. It just remains the same indefinitely. It doesn't actually happen in real populations because, of course, they're always changing, but Hardy-Weinberg is kind of like a theoretical thing.
What's the evidence that you can observe in year 0? We say there's no heterozygous like YD alleles.
Yeah, I'm not sure for number 2.
Well, 3. What are the 5 things that cause the population to not be in equilibrium? So the evolutionary changes, right?
Yeah, so that would be mutation, gene flow, genetic drift, non-random mating, and natural selection.
I always said it was in equilibrium.
Yeah, I don't completely understand that question. But the next problem, I think, might just give us the answer. uh, but I don't know what the evidence is. I'm not really sure because, like you said, it is in equilibrium. So, like, there's nothing not causing it to be in equilibrium. Yeah, I don't fully understand, but we could just say natural selection and then hope that somebody else comes up with the evidence.
Hmm. There's just those 3 things that we talked about in class yesterday or the other day. You guys think it's directional?
Uh,
I'd have to look up the definitions again. Hold on.
Yeah, because I think like directional, like it's just like going in one direction. And destructive, like there's like a reason like for it that's disrupting the population to go that way. And then stabilizing, I think, would kind of put them about even. So like that directional will be my guess.
That sounds good to me.
yep, I would agree with that.
Unless there is something disrupting them that's making them be yellow. But I kind of just think we said it's natural selection, so just like yellows, they're just more dominant. That's it. Is that it? That's it. We can go back to the room.
So save the paper, and could you please share this paper with me later, please?
Yeah, I can email it to you.
Okay, just later. Thank you.
Do you guys want a copy too?
No, I'm good. I've been typing with you, so.
Yeah, same.
All right. See you guys in there.
See ya.
For some reason, I clicked instead of leave breakout room, I hit left meeting. So can you give me permission to record again?
Oh, yeah, because you came back in again. But at least you let yourself in from the waiting room.
Yeah, for some reason it allowed me to this time.
Oh, maybe that's because you were already in the meeting once already.
Maybe. I don't know. It
Maybe,
just popped up for me. Okay.
maybe, who knows?
I
Zoom.
don't know.
you
It's
know?
magic.
Yeah. All right, so you guys got all the way through the worksheet. I'm gonna call the other, the gang back. Does one of you who is here have a worksheet with answers written on it?
They can post it up. I think they could share it on their
They
screen.
could share it, okay. Because I think people, and that'll make it pretty quick to kind of, whatever you would call it, it's kind of scroll through it. All right, yeah, and I got a thumbs up from Desiree. Okay, I'm gonna go get my other groups back here. Looks like, oh yeah, this is room 4. I was talking with room 4. Oh, and room 1 is, they've been working on it, okay. All right, looks like we're pretty much back from our, yes, we're just waiting for Christina. So can I ask this question of, Abdul couldn't come to lecture today and he asked if there was someone who was recording who would be willing to like post it on Google Drive or whatever and share a link with him. Is there anyone who would be willing to do that for Abdul?
Yeah, I can
I
do
know
that.
he, Kat, you'll do that?
Yep.
Okay, great, thank you. And Ju will. Yeah, that's great. And you know he would do it for you. All right, so it looks like 3 out of 4 of the groups got all the way through the worksheet and or almost all the way. So let's get one of you to share the worksheet and we'll just kind of go through it. So let's see, who said they could share their screen of the worksheet? Was that Desiree? Okay. All right. Do you want to just talk us through it kind of quickly, and then if people have any questions, they're gonna like raise their hand, and we'll go. So maybe go through it quickly, and if anybody has a question or comment, raise your hand, and then we'll slow down and talk about it.
Okay. So the first one we got 0.9 for the Y allele and 0.1 for B. And the genotype we got 90% for YY, 0% YB, and 10% for BB. And then for yellow perch we got 9,000, and for blue we got 1,000.
All right, looks good. Looks good.
And then part 2, in the absence of the 5 things that cause evolutionary change, what will the population be like next year? And we got 0.9 again for Y and 0.1 for B. And then we got 81% homozygous YY, 18% heterozygous YB, and 1% BB. And we got 9,900 yellow and 100 blue.
Looks good. Looks good. That's what the group I was with got.
Yeah, and the next year, like everything's the same because nothing changes, but there's no evolutionary processes. And then it asks if it's in Hardy-Weinberg equilibrium or not. And we said yes, it conforms to the equation because there was no evolutionary change.
Oh, okay.
Yeah,
Now,
and
is that, okay. Now,
Oh.
is that what everybody, is that what other people got? So in the population in the very beginning with 90% homozygous A, Y, and 10% homozygous B, was that population in Hardy-Weinberg equilibrium? Let's take a quick vote. If you think it was in Hardy-Weinberg equilibrium in year 0, give me a check mark. If you think it wasn't in Hardy-Weinberg equilibrium in the first year, give me an X. I can't see my list of my tallies. I can only see Andali voting for some reason. Okay. No, nobody's saying no. And yet the population changed, right? Year 0, when you observed it, it was 90% homozygous Y and 10% homozygous B. A year later, the genotype frequencies were different. Yeah, okay. So, in fact, this population was not in Hardy-Weinberg equilibrium in that first generation. Because, and you know that for 2 reasons. First of all, you saw it change. Once you put on the Hardy-Weinberg equilibrium conditions, it changed. The frequency of the genotypes changed. But even without doing it for a second generation, you could tell that it wasn't, because the frequency of homozygous Y individuals was not the frequency of Y squared. In that very first generation, the frequency of the homozygous Y individuals, that frequency was not P squared. And the frequency of the homozygous B individuals was not Q squared. And the frequency of the heterozygous individuals was obviously not 2 times P times Q because it was 0, right? So if P is the frequency of one allele and Q is the frequency of the other allele. Yeah. So I hope that you can see that, right? I think a lot of times when you learn Hardy-Weinberg, your instructor probably starts with equilibrium. But in fact, basically no natural population is ever in Hardy-Weinberg equilibrium. I mean, I'm an ecologist, right? So I look at real populations in the real world and they're never in perfect Hardy-Weinberg equilibrium. And to us, the interesting question is not, is it in equilibrium or not? The question is almost always, why is it not? Because like real populations are never in equilibrium, right? Evolution is constantly happening. So to an ecologist, the interesting question is, okay, it's not in equilibrium, why? And I know that this is a different perspective from probably what you learned in a genetics class. I think in a genetics class, they just want you to learn Hardy-Weinberg equilibrium. They're not really thinking about the whole, how an ecologist uses it. They just want you to understand it in the first place. So now we're going to take it one step up. You understand Hardy-Weinberg equilibrium. Now let's look at real populations that aren't in equilibrium and ask this question. There are 5 things that can cause it to not be in equilibrium, which you have listed here correctly. Which one of those is likely to be working on this population? The one we observed in the beginning. Okay, so with that long preamble, I think it's entirely possible that every group would get a different answer to question 4. So we're going to start with Desiree's answer, and then I'm going to ask each group that got to this question what your answer was. So what did you guys think, Desiree? What did your group think, and why did you think that?
Yeah, we just went with natural selection because there's a dominant alleles like the Y allele, and that ends up being
Oh, okay.
the yellowfish.
Yeah. All right. Let's see what other groups maybe had an answer. Juice, Saul, and Sam, what did you guys think?
We chose natural selection as well.
Okay.
And
why did you think that?
we chose that because based on Tuesday's discussion, when you mentioned that mutations usually occur cell by cell, so they take time to cause evolution, and therefore we just thought the natural selection made the most sense.
Okay. All right. Let's see. How about Brianna, Sam, Tiffany, and James? What did you guys think?
We said gene flow. We just kind of made the assumption that fish migrate all the time from lake basins to basins, lakes to lakes, and then we also just kind of thought about like the human effect of people just throw fish in different lakes in different areas all the time, and that could cause a genetic variation then in a population.
Okay. All right. I think those are 2 good arguments. Hey, Desiree, can you scroll up to the top, back to the population you're starting with? Okay. All right. So when you look at the population that we started with, the 2 things that are really striking are there's no heterozygous individuals, and there are a lot more blue individuals than there would be at equilibrium. Like if you scroll down a little, can you scroll so we can see both first and second? Yeah, look at this. So when we observed the population, 10% of the population was blue. At equilibrium, only 1% is blue. Do you see that? In other words, there's way more homozygous blue fish than equilibrium would predict. That suggests, well, natural selection, like something is really favoring the blue fish. Now, so I would say that seems likely, and the evidence is that there's way more of the blue phenotype than there would be at equilibrium. Can you see that in these numbers? Or do I think that that's obvious, or do you see that also? Give me a little check mark if that kind of makes sense to you. I'm getting like, it makes sense to like 4 people. Okay. All right. Another to me likely possibility of those 5 is that it could be gene flow. Because there's no heterozygous individuals. So maybe this is 2 different groups of fish that have not actually been together long enough to produce heterozygous offspring. You know, maybe this is a mix of 9,000 yellowfish from one side of a lake and 1,000 bluefish from the other side of the lake, and maybe they've never actually been together before. And in that case, you would call it gene flow. It's really kind of like 2 populations. Or maybe it's one big population of yellowfish that 1,000 blue ones have just joined. Because there's no heterozygous ones. Now, it could also be natural selection against heterozygous individuals. There might be something hidden that's dysfunctional about being heterozygous, and so they all die or something. The fact that Y is a dominant allele doesn't mean anything really. All it really means is when you've got two different alleles, the Y allele is expressed. It doesn't mean that it's better or stronger, although the word dominant kind of implies that. It doesn't even mean that it's more abundant or more frequent, which is another thing the word dominant sometimes means. In genetics, dominant only means when it is heterozygous, that allele is expressed. So, and I will have to say that is a very common misconception. So if that sounds confusing to you, really think about it, because you may have got the wrong idea about what dominant means. So check your understanding of that word. Okay, so let's scroll a little bit down further. Thanks, Desiree. Let's see, we got next year. We keep going. And there's your evidence that it's not. And then the last question would be something like this. Let's imagine, yeah, keep going. Okay, and then the final question, this is almost like a bonus question, right? Because I thought some groups would have time to get this and some wouldn't. But this is the last important question is, consider the hypothesis that natural selection is favoring the the blue colored fish. That frequency, that 10% frequency is being maintained by natural selection. Okay, that's a hypothesis. I mean, it doesn't have to be true. I mean, it could be 10% blue fish just because of gene flow, right? But consider the hypothesis that it's 10% blue and that's being maintained by natural selection. In that case, three things must be true. In general terms, phenotypic variation, heritability that affect fitness. Different individuals have different phenotypes. Some are yellow, some are blue. Correct. Heritability, no. I mean, heritability, yes. And that would mean the yellow fish have yellow offspring, the blue fish have blue offspring. Yeah, it's simpler than what you say. And then it affects fitness. The blue perch have more offspring than the yellow ones. If natural selection is favoring the blue morph, then the blue morph has to produce more offspring. Does that make sense? And then what this group did was they combined both of those right there on the same lines, right? They've got the general things on the left, phenotypic variation that is inherited and affects fitness. Those are the general statements. And then over on the right hand side, they've got it spelled out how that applies to this particular example. Obviously, this is one of my favorite exam questions because, well, first of all, you have to have memorized the three things. That's kind of easy. But then the hard part is you have to really understand those three things to be able to apply them. So it's the kind of question where if you have a little bit of knowledge, you can get partial credit. But to get 100% of knowledge, you need actual upper division, actual understanding. Which is to say, like, part of this question is kind of easy, just memorize it. But then part of it is hard. So it's kind of like, you know, a question that's good for everybody. Okay, so that's our worksheet 1. And that's the first thing that I wanted to go over today, was to review these things and to give you some practice really using this knowledge on kind of realistic type questions. Do you have any questions about the Hardy-Weinberg or the evolution that we just covered? Because then we're going to take a break and come back onto a different topic. She said, if Hardy-Weinberg equilibrium is true, it'll apply only to the offspring and generations after that. Well, usually what we say is, you have to say when when did the conditions for Hardy-Weinberg equilibrium occur? Before or after this thing I'm describing? Thank you, Desiree. Thanks for sharing your group's work. I'm not sure, Ju, if that answered your question.
Wait, there
think a
was
lot
one,
of-
one more question at the end.
Sure, you have another question?
Oh,
Sorry?
there was. What about this one?
Oh yeah, is it directional, disruptive, or stabilizing? And if you scroll up to the question, it says the blue morph is being maintained by evolution by natural selection. So that means it's stabilizing selection. Natural selection is not, well, it's not increasing the frequency of the blue morph. If it's causing the blue morph to get more and more and more abundant, that's directional. If it's maintaining the blue morph, that sounds like stabilizing. But on the other hand, you could say, well, it's maintaining both the yellow and the blue. So that'd be disruptive. Yeah, maybe that question's not super clear because in this example I've given you, there's only two morphs. It's just yellow or blue, that's it. There's nothing in between. And usually when we talk about directional selection, we're talking about a quantitative character. So it would be like the whole spectrum of colors from blue to yellow. So maybe that wasn't actually a good question. It was maybe a little trickier than I meant. What I was thinking was, oh, it's stabilizing because it's maintaining the blue. But as I say this out loud, I realize you could probably interpret that question more than one way. So nah, not a good question. Does that answer your question? So directional selection is like making a population more and more and more something. Stabilizing selection is kind of maintaining it at some sort of intermediate characteristic. And disruptive selection is when natural selection kind of maintains two different two different levels of something, like maintaining both light ones and dark ones, but nothing in between. You know, maintaining both yellow ones and blue ones, but none of the colors in between. Or maintaining small ones and big ones, but no mid-sized ones. That would be disruptive selection. And it's always like on a graph where the characteristic is on the x-axis from, you know, whatever, small to large or light to dark, or, you know. So I think actually those terms don't really apply that well when you've only got two morphs. Yeah. Okay, well, it's time for a break. Let's take a 10-minute break. We'll come back and have another topic to review. So 10-minute break. Sure seemed like it went by quickly, didn't it? Yeah, beautiful day outside. All right, well, anyways, let's go on to our next topic. I hope that everybody is back. Yes, are you back? If you're back, give me a check mark or a hands up or confetti or I don't know, thumbs up. Yeah, confetti, yeah. Oh yeah, I like the confetti. It's like, oh, it's like a party in here. Oh, and look, Hannah's here. Hi, Hannah. Welcome. All right. Well, what we're going to do now is just talk about how to draw food web diagrams. We're going to talk about food webs in more detail in the second unit when we've learned more species on more levels of the food web. I'm going to show you how to draw, and then I'm going to give you a challenge. All right, so to draw a food web diagram, you start at the bottom. I'll draw it over here, and I'll give you some rules over here. So you start at the bottom of the diagram with the abiotic resources. Some people don't always do that, but I think it's pretty important to remember that light, for example, is the real base of the food web in most situations. But, you know, the light is the energy. And then that light is, that energy is turned into organic matter by the phytoplankton. Right? Phytoplankton, the algae, they use photosynthesis to convert this energy into food calories. So this level is often called the primary producers because they're the first ones to make food calories. You know, to make your, you know, yeah. Now, we haven't talked about really species of phytoplankton yet, so I guess I don't expect you to know any names yet. But you know the names of several zooplankton. The names that you need to know are in that PowerPoint. Bosmina, Daphnia, Caratella, Chaobarus. So phytoplankton are eaten by things like Daphnia and Bosmina. Daphnia and Bosmina are basically herbivores. They eat algae. They will also eat detritus if it's a small enough particle. And they will eat bacteria. If there's bacteria in the water, like little unicellular things, they sweep into their maw and eat. But for the purposes of this food web, in which most of the food is coming from phytoplankton, I'm going to leave out those extra little details, like the detritus and the bacteria. I'm just going to leave them out because they aren't a big part of the diet. So we've got the Daphnia and the Bosmina. They eat the same thing, so they're on the same level of the food chain. The food web, if they're on the same level of the food chain, you put them on the same level in the diagram. Now the Daphnia and the Bosmina are going to get eaten by sunfish. So let's say that they get eaten by bluegills, which are a kind of sunfish. Okay, and let's just keep it simple. I'll just only have the bluegills here. And you'll notice bluegills eat Daphnia and Bosmina, so the bluegills are above them in the diagram. You always put things above what they eat. If a species eats more than one thing, so you could put it on two different levels, you put it on its highest level. So bluegills eat Daphnia and Bosmina, but so do baby walleye, right? But adult walleye will eat the bluegills. So I'm going to put walleye here because when the walleye are adults, they eat the bluegill. So they have to go above the bluegills in the diagram. But when walleye are young and they're small, they actually eat Daphnia too. So I'm going to draw a line like this. So because the walleye eat on two different levels, you put them on their highest level. And then, well, I want to put muskie into this diagram. Where do muskellunge belong on this diagram?
Above the walleye?
Above the walleye, yeah. So we're going to have the muskies up here. Now, it's true that adult muskie can eat adult walleye, and they can especially eat juvenile walleye. But muskie will also eat bluegills. So we're going to draw another arrow here. And I suppose you could even say that when they're juveniles, they eat that. When they're like really young, when the muskie are just muskie fry, they eat Daphnia too. Maybe in this lake, the muskie are stocked. And so they're never in the lake as fry. So maybe in this particular lake, I'm not even going to connect them to the zooplankton level. And you know that muskie occur in about 100 lakes in Minnesota, like whatever, 91. And in 2/3 of those lakes, they don't occur naturally. They're only there because humans stock them there. So that's pretty reasonable for me to say, well, these muskie in this lake are never fry. They're stocked as fingerlings, at which point they're already eating, I swear, other fish. They grow so fast. Okay, so here are the rules for drawing a good food web diagram. First of all, start at the bottom. Right? With abiotic resources. Or if you don't include the abiotic resources, then the bottom of the food web is the primary producers. Right? I don't mean that these two things are the same thing by using that word, or what I mean is some people start at the bottom with abiotic resources. Some people, or some purposes, start at the bottom with the phytoplankton. Okay, but in any case, you got to start at the bottom. You put each species or taxonomic group or whatever, however you're grouping these things, you put each member here above what it eats. So you put each species or whatever taxonomic resolution you're using and you connect each thing to what it eats with a line. Now, you don't actually need to include the arrowheads if everything is above what it eats. Then the arrowhead is actually unnecessary. You know what is the flow of energy. It's always up, right? So arrowheads are optional. But if you do use arrowheads, put a head on the arrow, you must be consistent. You know, don't use arrowheads for some things and not for other things. If you use them for everything. If, like I've shown, the arrowheads point up, you know, the pathway that energy is taking, then they always must point up the direction the energy is taking. Now, you can, sometimes people do food webs with the arrows pointing down. And what they're showing in that case is control, impact. They're showing the role of predation on the prey. Sometimes you make a food web to show the flow of energy. Sometimes, essentially, you're showing the impact of the prey upon the predator. Like, if a thing is not an important part of the diet, you don't include a line. So I said, you know, maybe the muskies do eat a little bit of Daphnia. Well, maybe I didn't say that, but I mean, adult muskie probably eat some Daphnia. I mean, they're swimming around in the water with their mouths open. They probably do eat some Daphnia. But the Daphnia are not an important part of the diet of the muskie. And so I didn't draw a line. Well, if you wanted to show how complicated the food web was, and that was your purpose in drawing the diagram, then you probably would include that line. So you got to know why you were drawing the diagram, and only include the lines and the species that are important given what your purpose is, right? So you're gonna, essentially, it goes without saying, you need to know why you're drawing the food web and include only the taxa, know the species and the lines that are important. So, you know, if I'm trying to show how complicated everything is, I would include every possible line, even if it's just a tiny little part of somebody's diet. But if I'm really trying to show, you know, how important each individual species is and to whom, I wouldn't. Like, if I wanted to show what would happen if I removed the bosmina, you know, what would happen if I took the bosmina out of this lake? Then I don't want to have every possible line on the diagram. I want to have only the important ones. And then I would see, well, gosh, you know, if I remove the bosmina, there's going to be more food for the Daphnia. And that might be a good thing for the Daphnia. But there's going to be less food for the bluegills. So that might be bad for the bluegills. Of course, bad for the bluegills, well, that might just mean more predation on the Daphnia. So that might be bad for the Daphnia. Well, the point is, if I had even more lines going on here, you know, like a little line up to the walleye and a little line up to, it'd be harder to figure out what is already a hard thing to figure out. So know why you are drawing your food web and include only those taxa and those lines that are important. And that kind of leads to the final rule, which is to say, combine or divide taxa, you know, to whatever degree it is useful. Now, what I mean by combining taxa is I just combined all the different kinds of algae here and called them phytoplankton. For my purposes in drawing this diagram, I didn't need to break it out into individual species. But I definitely divided the Daphnia and the bosmina apart from each other because I wanted to ask a question like, well, what if I got rid of the bosmina? And what about dividing? You know, the walleye occupy different levels of the food chain depending on their age. When they're just a few weeks old, they're fry, they actually function as zooplankton. At the end of the first year, if they're big enough, then, you know, then they are, well, actually, no, come to think of it, even after the first few weeks, they're big enough that they begin to eat zooplankton, and then they are, would be at this level as planktivores. By the end of the first year, if things went well, they're going to be at this level eating other fish. Maybe not bluegills, but, you know, minnows and the young of other species in the springtime. 1-year-old walleye are effectively piscivores. And in fact, after a couple of years, they're so big, the walleye are, that they're eating the fingerling walleye, which are eating the walleye fry, and so on. You might decide that's really important, in which case you're going to divide the walleye into different subgroups, stages, fry, fingerlings, young, adult, depending on your purpose. Now, a species like this, where do I got to put this? Oh, I missed one point, which is to say, what do you do with something that occupies more than one food level? You put it at its highest level. I don't have enough room here. I'm going to write it up at the top. Here's my asterisk. Chronologically speaking, this note's going to go on the bottom or maybe in the middle, but I'm going to put it up here because I have room. And that is to say, put omnivores at their highest level. I'm not sure you can read that. There's too much of glare on the screen here. This says, put omnivores at their highest level. Now, an omnivore is a species that feeds on more than one trophic level. The muskie eats piscivores, and the muskie eat planktivores. Therefore, the muskie is an omnivore. Now, this is not how the word omnivore is used in sort of common English. In sort of common English, how do we define omnivore? Like if you were talking to your, I don't know, your mom or your sister or your brother or somebody, what do most people think an omnivore is? Do you want to put it in the chat? I believe this is what most people think. So maybe we could just check that. What does the word omnivore? Yeah. Yeah. So kind of your sort of ordinary people definition of omnivore is something that eats both plants and animals. But in food web theory, that's only one kind of omnivore. In food web theory, an omnivore is anything that eats on two different levels. Now, obviously, bluegills, if they ate Daphnia and phytoplankton, would be omnivores. But in aquatic food webs, all of the animals, okay, whatever. Lots of animals are omnivores in aquatic food chains. Most of the animals are carnivores and simultaneously omnivores. So the muskie is an omnivore because it eats both walleye and bluegill. The walleye are omnivores because they eat both bluegills and zooplankton. Yeah. And so we put each of them at their highest level on the food chain, and then you just be sure to connect it to what it eats. Okay, so that was one technical term, that's omnivore. The other technical term is a guild. And when we say that we're going to combine species that occupy exactly the same place with the same connections, we call it a guild, which is, I got one definition up there, I'm going to put the other one down here. And this is the definition. I'm not sure you can read that, but a guild is a group of taxa, and taxa is a word that kind of means like species, but it's more general. It could be like species, or it could be like genus, or it could be, it's some kind of taxonomic group. So a guild is a group of taxa that occupy the same place and connections in a food web. So here's a guild, the phytoplankton. That's a whole bunch of different species. They're all primary producers with one arrow connecting them to the abiotic resources. And they are all eaten by both Daphnia and Bosmina. And I just grouped them together because there wasn't any point in spelling out a whole bunch of different species when, for my purposes, they all occupy the same place with the same connections in the food web. Now, I could have combined the Daphnia and the Bosmina, but they're not a guild in this food web because I said the Daphnia get eaten by the walleye, but the Bosmina don't. So they're on the same level, but they are not a guild because they don't have the same connections to the other things. Really similar, but not exactly. Two different things eat the Daphnia in this food web, but only one thing eats the Bosmina. So the Daphnia and the Bosmina, they're both zooplankton, but for the purposes of this food web, they are not a guild. The word guild comes from bird biologists who there's a lot of diversity of birds in the world, and a lot of times they just wanted to be able to say, that the insect-eating birds, the insectivores, the fruit-eating birds, the frugivores, and just so they kind of invented this concept of guild, or brought the word guild into food web ecology. All right, so this is an example, and these are the rules for drawing a good food web. In this class so far, you have learned the names of a bunch of different fish. You remember we had a lecture where I showed you the top 10 fish you need to know? Yeah, those were the top 10 fish you need to know in Minnesota. And then I showed you a lecture on zooplankton. I don't think I showed you 10. I think it was less than 10, but I showed you, Vorticella and Caratella and Daphnia and Bosmina and Chidoris. And then I showed you Chiabrus, the phantom midge larva. And then I showed you some walleye larvae. I might have missed one. But anyway, so those are like the zooplankton taxa you need to know. You need to know what they are. You need to be able to maybe picture them, know something about them, know where they fit in a food web. So what I'm going to do now is I'm going to ask you to draw a food web following these rules and using the set of species listed on the worksheet. And then I put in 2 questions. What were the 2 questions? Oh, top down and bottom up. Yeah. So one of the reasons that you draw a food web like this, with abiotic resources at the bottom and eaters above what they eat, is so we can use these terms top down and bottom up. If you wanted to increase the abundance of walleye in a lake using a bottom up strategy, you would probably fertilize the lake with more phosphorus, because the overall phosphorus concentration in lakes is very strongly correlated to the productivity of the phytoplankton. The productivity of the phytoplankton, of course, influences how fast the zooplankton populations can grow, which influences, the planktivorous fish populations, which is the food for the walleye. So a bottom-up strategy for increasing the walleye in a production of a lake is to fertilize the lake. And there are ultra oligotrophic lakes, like in the Rocky Mountains, where people have deliberately fertilized them in order to increase game fish production. I mean, that seems kind of amazing to us in the land of 10,000 eutrophic lakes. You know, we're always trying to reduce the eutrophication, but I mean, there are places where the opposite is true. Now, what if you wanted a top-down strategy for increasing walleye? What would you do? Here's our lake, here's our food web. What would be a top-down approach for increasing the walleye productivity of this lake? Give me a suggestion, put it in the chat. What would you do to increase a top-down approach for increasing the walleye? Got 2 suggestions? What do you think? Top-down approach for increasing the walleye. What would you do? Okay, I've had two people who have had some ideas. Let's see some more ideas. Come on, Bree, what would you do? You want more walleye in the lake? You're going to take a top-down approach.
What was that? Sorry.
You want to increase the walleye population in the lake, and you want to take a top-down approach.
Yes.
So what would you, okay, I think Bree wasn't listening, so I want somebody else to give me an idea.
You would have to increase the food source.
No.
No.
No, good try though. Thanks for trying. I'm going to pick on you because I think you weren't paying attention. Is that okay?
That's fine.
I don't mean to hurt your feelings or anything.
You didn't hurt my feelings. I spaced out.
You didn't know I was going to call on you, did you? All right, we're getting some answers coming in here. And you know what? I'm gonna tell Kat that she's right? And somebody else was right too, but it scrolled off the screen. Right? A bottom-up approach means in order to do something to this population, you're going to manipulate the food web beneath it. So fertilizing the lake, stocking it with more zooplankton, or stocking it with bluegills, those are all bottom-up approaches to influencing the walleye. A top-down approach is to manipulate the food web above the species. Therefore, reduce the muskie. If you reduce the muskie, you reduce predation on the walleye, and maybe the walleye population would increase as a result. So one thing you need to know about food webs is you need to draw them this way so that you can talk about a top-down effect or a bottom-up effect, a top-down approach or a bottom-up approach. And it could be regarding the fish population that you want to change. It could be about the phytoplankton population. Or people don't usually want to manipulate the zooplankton in and of itself. People usually care either about the fish or about the algae. That's just the way, people are. All right, so what I'm going to do is I got some slightly modified discussion groups because we have a slightly different set of people here. There is a handout posted called Food Webs in the same place as the other one. And it has your next assignment. So I'm going to send you into your breakout rooms to work on this assignment, which is basically draw a food web and then predict 2 things. Uh-oh, where did we go? Hey, Zoom. Okay, send some people there. All right. We've got some rooms to go to. Bree's going to go to room 1 and Hannah Hannah is now here. I guess I'm going to send Hannah to room 3.
So what was the question she asked us? I was listening, but I didn't really understand what she was asking.
She created another worksheet and she wants us to make a food web.
Oh, okay. Yeah, I have to print that.
Are we supposed to do the food web or the population one?
The food
The
web.
food web.
Does anyone want to share their screen?
Desiree, I can. I feel like you've been doing notes, but we've been in a multiple groups together before, so.
Yeah, it's like in all my classes, I'm just like, I volunteer, tribute, why not? It's like then I know I have the notes because I did the thing.
Right.
Yeah.
Bless you.
It was the noise.
Did I sneeze? Who was that?
Oh, there was a noise when you came on, so I made a noise.
Oh,
What was that?
oh, this is like a super big group, so I'm gonna steal Kat and send her to a different room. Is that okay?
Yeah, that's okay.
Is that okay? Okay.
And then, do you both think that do you think it would be walleye juveniles would be on top of that? And
I
then
want
and then
to
do you
say
think,
it's maybe
oh
the
sorry.
fries.
Oh, sorry, fry. So
Yeah.
so it would be fry, and then I have a feeling sunfish and walleye juveniles would be on the same level.
Yeah, I think they definitely both eat the walleye fries, so
Perfect. So
yeah.
yeah, so we'll do walleye fry its own. And then above that one, we're gonna put walleye, walleye juveniles and sunfish. Those will be on the same level. I have no idea how this exam is gonna go on Thursday. I just have no idea.
Yeah.
I just have no idea how hyper specific some of these questions are gonna be. It's like, do I need to rewatch 3 weeks of lectures and then just like double check all of my notes, or do I just need to go through the study guides of what I've read in class?
I mean, the study guides are pretty damn specific in this class, so
I know. So hopefully she sticks with those. But yeah, I'm glad that she kind of told us that we do need to be able to identify these species, because I did not really go over that yet. So it'll be nice to try to memorize all these zooplankton species. That'll be fun. And And then the last one would just be walleye adults all the way up top. Can you put arrows in on
Yes.
And then I think we should probably include an arrow showing the walleye fry going up to the walleye adults too. Looks pretty good to me. All right, now we need to answer those two questions. Propose a bottom-up method for increasing the abundance of sunfish in this lake and explain how it would work. So if we wanted to increase the sunfish population from a bottom-up approach,
We'd increase their food.
yeah. So I guess kind of directly we could just increase maybe make the lake a little more eutrophic, add more phosphorus or nutrients to have the phytoplankton feed on.
Yeah.
And then more of a phytoplankton will increase the amount of abundance for the walleye fries, and then from there more sunfish could eat on the little guys, and then also the phyto. Do you think sunfish directly eat phytoplankton too?
I don't think so. They might eat zooplankton, but I don't even really think that.
Okay.
They're a little small for sunnies.
I'm just trying to think if maybe we should add arrows from the zooplankton species to the sunfish.
I don't think so. A sunny's a little bit big for those zooplankton there.
Cool. All right, so we got that, and then a top down, increasing the abundance of sunfish.
Decrease the predators, decrease the walleye.
Yeah, just decrease the walleye population.
Yeah.
Don't worry, I was paying attention this time.
I know, I can't tell, right? Because everybody just has a picture or a name. So yeah, sorry for picking on you.
No, it's fine. I think it's kind of funny. It's like, how did you know? Because it's like I'm kind of just kind of staring off to space. So
Yeah, I know, it's tough. It's tough. All right, it looks like we got everybody back. Almost everybody back. Just a couple more people from Supergroup 3. I'm going to ask room 1, which would be Bree, Sam, Saul, and Ju, to put up there to share with us your results, if your note taker saved it. All right. You want to just walk us through this?
Yeah, so at the bottom of the food web are the phytoplankton. And then these 4 right here, zooplankton, the daphnia, walleye fry, caratella, and the bosmina. I think earlier when you mentioned that walleye juveniles or walleyes don't eat bosmina, so we don't have an area
Oh.
that. Yeah, and then we put these 2 on the same level, but then we're debating as to, we're wondering if walleye juveniles actually feed on sunfish. So there's a question mark there.
Mhm.
And then we have the walleyes at the top of the food chain here.
All right, that looks really good. Yeah, you weren't given a ton of information, and you could have drawn this a little differently based on those things that you've mentioned. I was thinking that this was in, say, April or May, so that you have walleye fry in the lake. And then the walleye juveniles are 1 year old. And then adults really means in the breeding population. So that's 3, 4, 5 years old and above. And I was thinking no juveniles in between, because that's that's complicated, as you've indicated. Where would the juveniles eat sunfish or not? I think that the 1 year old walleye are not really big enough to eat sunfish. But that 2, 3, 4 year old juveniles might be, Bosmina are, generally speaking, smaller than Daphnia. They're similar, but smaller typically. So that's so that, yeah, maybe they get eaten differently. And yeah, it's typical that the planktivores eat the bigger ones first, because fish, these fish anyways, are visual. They'll see the little speck and they'll eat it. They'll see the daphnia and they'll eat it. But the bosmina are smaller and it's harder for them to see. So there is, at some point, some important differences between them. So you may, in your other, people in other groups may have had slightly different food webs, depending on whether you wanted to incorporate that detail or not. The description you're given for this assignment was very brief, and I tried to keep it brief. Now, one option you might have thought about would be, maybe you would have thought, well, these things all get eaten by the same things, in which case maybe you would have called this whole level, by a guild name like zooplankton. Or, to be more realistic about it, you might have said, if I could do this quickly, you might have said, these are big zooplankton and they get eaten by walleye and sunfish. And these Caratella and Bosmina are small, if I said that correctly, small sunfish, small zooplankton. So you might have divided this into two guilds, the small zooplankton and big zoops. So you might have decided to use guilds. If you used guilds, they would look something like that. Now, there are no omnivores in this list, in this food web diagram as it is currently drawn, because the walleye here have been divided into 3 different size classes, which occupy 3 different places. If you lumped the walleye all together, you'd have to call it an omnivore. But in this case, you don't, because you don't have walleye. You have walleye adults, walleye juvenile, and walleye fry. Maybe that's not completely clear, but you wouldn't necessarily call the walleye adults in this diagram are not omnivores. The walleye juvenile in the diagram are not omnivores. The walleye fry in the diagram are not omnivores. But I suppose the walleye as a whole are omnivores. And that's what I wanted to say about this diagram. Nice job. I think this is, pretty much exactly what I was hoping you would draw. Well, how about answers to the questions? What did you come up with?
They were very similar to the answers that we came up with as a class for the walleye earlier. So for bottom up, just add phosphorus, which kind of causes lake eutrophication and resulting more nutrients for the other trophic levels.
Okay.
And then for the top down, again, we didn't focus generally on the 1-year juveniles. And so we said that if we remove the juveniles and the adult walleyes, there'll be less predation and competition for the sunfishes.
Yeah, okay, so if you scroll back up to your diagram. Yeah, so, oh, where'd the adults go?
I don't know.
Oh my gosh, the adults vanished. It's like, Peter Pan or something. Okay, so the adult walleye are above the sunfish. So a top-down approach would be to remove the adult walleye. Removing something on the same trophic level is not a top-down effect. It is biomanipulation. It is manipulating the food web, but if it's not manipulating the food web above your target species, then it's not top-down. Manipulating the phytoplankton or manipulating the zooplankton, yeah, that would be a bottom-up effect. Okay, so these are, good answers to both questions. Maybe with that, nice job, Bree, with that modification. Okay, well, I just want to wrap it up today. Thank you for sharing your screen. And thanks everybody for working on these worksheets. I kind of actually sort of liked the way that went. I thought that people were really, it seemed pretty productive to me. So maybe mixed with lecture. Bree says, yeah, confetti. So next Tuesday, we will have, what are we doing on Tuesday? We're gonna have class. We will discuss the article by Hairston. So please read the article. It's only 1 page long. It's not hard to read. Okay, there's like some gnarly statistics right there in the middle, but everybody should be able to read at least some of that article and understand some of it. So prepare for class on Tuesday by reading that article. Also, on Tuesday, we will review. I have one more worksheet in mind. I might come up with another one. I like that. And the idea with the worksheets is to give you actual practice working on potential exam questions of the bigger type. There will also be little questions questions on the midterm exam, and those will be drawn largely from the reading questions. So look through, as you study the reading questions, look through your lecture notes. I can't actually see if you're taking notes. If it was a classroom, I could see. I assume that you're taking notes the same way you would in a real classroom class. And your notes are a record of what we went over in class and what I emphasize. So look through your lecture notes to see. And if you have any questions you want me to go over or to explain in a review session, there is a survey on D2L called, I forget what, Review Q&A. And in that survey is a place for you to enter your top 3 questions that you want me to go over during a review session. Type out the whole question. Don't just write like the number, because write the question that you want me to answer. And in the review Q&A session, I will go through the questions that people have submitted before class, and I will answer the ones that I think you really need to know. There is a quiz on this week's lecture material. It will open tomorrow at 1 p.m., which is when I imagine that they open each week, but this week it really will open at 1. It's a typical, 10 or 15 minutes, 8 or 9 multiple choice, I've forgotten already, 8 or 9 multiple choice questions, and then 1 write out the answer question. And that will raise the total number of quizzes to 12. Each person will still count their 10 best quizzes. So this is sort of like a bonus quiz. It gives you an extra quiz to drop. I think that's what I wanted to say. Okay, questions, June.
Oh, can we, so for those review sessions, can we ask you quiz questions as well?
Yeah, if you have a quiz question that you got wrong, theoretically the quizzes are set up so that if you get a question wrong, after the deadline for the quiz, you can see the question. That's supposedly the way that it's set up. Yeah.
You can see the question, you just don't know what the answer is.
Yeah. And that's supposed to help you learn. The exam will be on campus, face-to-face. Well, not face-to-face exactly, but on campus. I'm pretty sure that it will be in SEC 115, which is the big room in the Science Education Center on Thursday at 1 o'clock. Desiree says, what about today's lab? I think what we're going to do is I have to figure it out. I love this lab, so I think we will do this lab in the second unit. We won't be talking about zooplankton anymore, but I still think it's worth doing. So I think that we will do the lab that was planned, but we're going to do it a little later. James.
Hey, I just have D2L open right now. I don't see the article posted yet that we're supposed to read.
You're kidding.
Yeah, I don't see that in the reading, and the survey that you mentioned, I don't see that up as well either.
Oh, well then. All right, D2L tends to make things invisible unless the instructor tells.
Other professors have told me in the past there's a lot of boxes you need to click, and if you miss one, it could just kind of screw the whole posting thing.
Yeah, and here they are. In fact, both of those things are posted, but they are invisible in D2L's words. So you should be able to see both of them now.
Okay, perfect.
Thank you for asking. Yes,
yeah.
Of course. I saw them.
Yeah,
other questions?
They just popped up now. Perfect.
Good. All right, other questions? Checking my chat here. All good. So let's see, the first exam is next Thursday. Tuesday, we will do article and review. There is a quiz that's due on Monday, a typical quiz. I graded the lab reports, and if you read your lab report grading and you're like, oh my God, I'm not, I can't believe I made the same mistake twice, or, you know, whatever, it's okay. I have put off the deadline for lab 2, which was originally due today at 1 o'clock. That deadline's been pushed until Sunday at midnight, just to give people a little extra time in case they wanted to change things. Ju, question.
Oh yeah, so if we already submitted our report, can we resubmit it if we want?
Yeah, just resubmit it. You can put a new one into the same Dropbox. D2L just labels it with the date you uploaded it. Unless you tell me otherwise, I will just read the newer. So if you submit 2 copies, I just read the newer one. I just ignore the older one. That's a good question. Other questions? All right, well, you know, I still feel bad that we didn't have lab today. I would really like to have been outside. Those of you who are here, do you want to just like give me like just a quick whether you were happy or whether you were sad about not having lab today? You want to just give me a quick reaction, like a check mark or a smiley face for happy, an oh, or a sad face or whatever you get for unhappy? Was it a good decision? 2 of you were sad. 3 of you were sad. 4 or 5. Oh, everybody was sad. It was a little cold, says Keenan. Yeah. I was really worried that somebody would get frostbite or somebody would get, you know, I don't know, whatever. This was good. And the weather's going to be awesome starting the week after next. Yeah, traffic was terrible this morning. See, yeah, and the review's helpful. I really think the review will help people who are here. So, all right, and that's what I've got for you today. Thank you to Ju and Kat for sharing your recording with Abdul. And I will see all of you next Tuesday, and some of you I will see on Monday, you know, and have a nice weekend.
Thank you, you too.
Yeah, stay warm.
Virginia, do you have a moment
I
when everyone
do.
leaves?